Welcome friends, welcome to another video from Generalized Biology, and in this video we will discuss about bacterial transduction method. What is meant by bacterial transduction? Bacterial transduction is the method in which gene transfer from a donor bacterial cell to the recipient bacterial cell takes place by the way of bacteriophage. Means bacteriophage play an important role in transfer of genetic material from donor bacterial cell to the recipient bacterial cell. We know that in the transformation, DNA is transferred, means gene is transferred directly from donor bacterial cell to the recipient bacterial cell. But in this method, in the transduction, the DNA is carried by the bacteriophage. Means bacteriophage play a very important role in transfer of genetic material. Or in transduction, DNA is transferred from cell to cell through the agency of viruses. Viruses nothing but the bacteriophages. Okay. Now let us discuss about types of bacterial transduction. Bacterial transduction is of two types: generalized transduction and second one is specialized transduction. What is meant by generalized transduction? It is the type of transduction in which potentially any donor bacterial gene can be transferred. Okay, as name indicate, generalized means there is no restriction to transfer the gene from the whole DNA, means that any general gene is transferred in the generalized transduction. Now let us move towards the second type, that is the specialized transduction. In specialized transduction, the transduction in which only certain donor genes can be transferred. As name indicate, that specialized, only a restricted gene is transferred from donor bacterial cell to the recipient bacterial cell in the specialized transduction. Types of bacteriophage. Bacteriophage is mainly of two types. First one is virulent phage and second one is temperate phage. Virulent phage are those phages which can capable of causing infection and also the destruction and death of bacterial cell. Okay, means that commonly we can say that the virulent phages follow the lytic cycle, and the example of virulent phage is the T4 phage. Second type is the temperate phage. Temperate phage does not cause destructive infection to the bacterial cell. Means temperate phage does not causing destruction to the bacterial cell. They does not cause the lysis of bacterial cell. Instead, the temperate phage containing their own DNA is incorporated with the bacterial cell DNA and forms the complex. Okay, and temperate phages following the lysogenic cycle. The example of temperate phages are lambda phage. Now let us discuss about generalized transduction. In the generalized transduction, virulent phages which are containing the phage DNA. Now virulent phages are binds to the surface of bacterial cell and infecting to it and releasing their DNA inside the bacterial cell. After releasing the DNA inside the bacterial cell, they can cause the breakdown of the bacterial DNA. Okay, and the replication of virulent DNA is now starting, and alternatively the new phage particles are also starts to synthesize, and during this packaging of new phage particle, the bacterial DNA is packed inside the virulent phage bacteriophage mistakenly, and this bacteriophage is infecting the another bacterial cell known as recipient cell and releasing its DNA inside it, and this donor DNA is now form the complex. It is incorporated with the recipient cell bacterial DNA. Okay, and in this manner the new cell is formed with the new recombination. Now let us discuss second type, that is the specialized transduction. In specialized transduction, the bacteriophage is temperate phage, and the example of temperate phage is lambda phage containing its DNA known as virulent DNA, and this DNA is now inserting in the bacterial cell, bacterial donor cell, and forms the complex with the donor bacterial DNA. Okay, this is the complex forming, and after some time it takes the some part of the some gene of the special gene of the bacterial DNA and with the virulent DNA. Okay, now from this complex the new phage particles in the next step
starts to synthesize with the replication. So many of the new phage particles are starts to synthesize, and these bacteriophages, new phages infecting the other recipient cell bacteria and releasing its complex donor DNA and virulent phage DNA and forming new complex known as transduced DNA. Okay, so this is the new recombination transduced DNA containing virulent DNA, donor cell DNA and recipient cell DNA. Okay,